This video is going to review what we completed in class on Wednesday, September 23rd and Thursday, September 24th for science. So taking a look at the agenda, we are learning to provide evidence that living things are made of cells and to develop and use a model to describe the function of a cell and its parts. And so we started off with the first learning activity, which is sharing our do-for diagrams of our cell cities. And so we projected different iPad screens of any students that wanted to share and kind of looked at their cities. And the whole point was to see the different ways that a cell city could be set up and to show how cell parts work together in a cell to make the cell function as a whole. And each of those cell parts or organelles have its own particular function, its own job, and they all work together to have a healthy cell. Just like in a city, there's different parts in a city. They all have certain functions that all work together to have a functioning city. And so we kind of related it to that. Then we completed our Do For Cell Architect Exit Slip, which was our second learning activity. And then we went and kind of reviewed all the activities we've done in 1.1 cells, since we're getting ready for our unit assessment. So let's take a look just at what we've done so far. So going back to the beginning, we always want to refer back to our cover page where we have our resources available, the learning activities that we completed, and you had an exit slip for each of those learning activities. And you can start to think, what do you need to do to prepare for this assessment? So you can look over the activities we did, your exit slips, the feedback I gave. You can look at the STEMscopedia reading, and you can also do those STEMscopes online practice activities, which again aren't a graded thing. They are just for you to help study, and there's practice tests and other kinds of things in there that once you complete and turn in, you will be able to see all the correct answers and you can self-check, and it's just another way to help you study. And again, nothing is going to be a grade in home access on there. It's just for you to help you learn and help study and prepare. And so that's what the STEMscopes online is. There's many activities in there. You don't have to do them all, but ideally you would do some, make sure you're good and you're getting the right answers and you feel confident, and then you're good. Some units you'll find might be a little harder than others, and you'll want to do some more of those review activities. Then in other units you might find you really understand quickly and easily, and you're good, so you won't need to do so many. All right, so the first thing we did is we always start with our question, our investigative phenomenon. In this case, it's, are viruses classified as living or non-living? And so first we had to learn what makes something living versus non-living. And then we have to learn still a little bit more about what viruses are and how are they, what makes them up to know if it's living or not. So we did do one, which was all about determining organisms, whether they're unicellular or multicellular. And then we did an activity where we collected evidence that living things are made of cells. So we looked at these microscopic images, which if we were physically in school, you would get to use a microscope and look at these things. But that's kind of what you would see under the microscope anyways. And you had to look to see that the leaf and the human blood had cells and the water and sand did not. And so you were able to determine that if you saw cells, you know something's living. All right, then we went to do 3, and you learned about some of the functions that plants, animals, and cells can have. And so it gives you a lot of items, and you didn't have to know what all these things are because it gives you a description. But it's just trying to expose you to different parts of plants, animals, and cells and what they do so that you can see that they might have different names and do things differently, but they have kind of the same main function. Whether it's from plants, animals, and cells. And we saw that many of these letters, these functions overlapped in our table. And then for do 4, you had to research some of the cell parts or organelles and find out what their main function or job was, and then relate that to parts of a city. And so we filled in this data table with the different parts of the cell and kind of the function. We looked up the function, did some research to see what city part it seemed to kind of match with. And we saw that the cell parts, each had their own function, just like city parts had their own function or their own job. But regardless, all of those parts work together to make a healthy cell or a healthy city. And that's what you learned in that activity. All right, so those are the main learning activities we did. Don't forget, we also have those STEMscopedias, which is like our textbook, and those should be in Notability. And you have the STEMscopes online also to practice and help review for your assessment. All right, so going back to the agenda. After that, we did watch the video on what is a virus so that we could really find out exactly what a virus is so we can apply our learning about cells to know if a virus is living or not. And so you should probably pause this video and then watch the what is virus video and then come back. So once you've watched the video, you'll see that they basically do come out and tell you that viruses are classified as non-living. And the reason is we've learned that cells equal living, that you have to have cells. That's the smallest part of a living thing. And anything smaller or that doesn't have all those parts isn't going to be classified as living. So a virus is really just some genetic material inside a protein kind of coating, and it's not really all that complex. It doesn't have all those organelles that a cell has, and so since a virus is not made up of cells, a virus is not living. And lastly, for the to-do, you would update your investigative phenomena after instruction column. So we did that together, kind of with what we learned in the video. So you would look at and fill in this third column. So we've already done the before instruction, the during, and then this is the after. And so now you know the answer from what we've learned in class. All right, and then for the to-do, you just have to review and get ready for your assessment. So if you had class on Wednesday, you're a blue day, your test is going to be on Monday. And if you're a green day, your test is on Friday, and it's going to be on 1.1 cells. And I did put a note about STEMscopes just to remind you that that's available, how to get your login information, that the activities are used as a study tool. You get to choose which activities you feel will help you prepare the most. You don't have to do all of the activities. And once an activity is submitted, you'll see the correct answers and you can self-check your work to see how you're doing. All right, that's it for what we did today.